main target is this hemicelluloses. Cellulose is also our target, but basically now is the hemicellulose is our target. So this hemicellulose has been found in a lot of biological activities. Moreover, they are also used as a nanotechnology. Professor Katarine was lecturing. I wanted to ask some questions, but in paucity of time I couldn't ask. But this also has been found to used by the nanotechnologists nowadays. This hemicelluloses. They also have to be have gastroprotective activity, pharmacological activity also they has, and they are also being used in the pulp and paper industry to enhance the quality of the paper. So we are basically interested in hemicelluloses, but we are also interested in hemicelluloses because of their potentiality to produce the bioethanol. Because if you see the petroleum products, the crude oil is going to be extinct one day or the other. So we have to have alternative resources of energy. So this cellulose, as long as sun is there, plants will be grown, and then this cellulose can be converted to the bioethanol. So we are also interested in this one also, but now our main challenge is over how to extract the hemicelluloses, better quality and better yield from the waste of the sugar industry. Well, so let me go through some of the statistics of the sugarcane bagasse. Nearly 54 million tons of sugarcane bagasse is produced annually. As I am from Tanzania, representing the University of Dodoma, so therefore we are more concentrated on this Tanzanian sugarcane. So in Tanzania, nearly 1,700 hectares of sugarcane is farmed. Out of that, 1.3 million tons of sugarcane is produced, and this bagasse produced from that production of sugarcane is 455,000 tons. This is annual production in Tanzania, which is a huge amount, of waste that does the sugar industry generating. And surprisingly, this is very 80% of this one, this bagasse that is produced, is burnt by the sugar industry because they don't have that biorefinery technology. They don't have that technology to extract these important compounds like celluloses and lignin. And therefore, what they do in order to dispose the waste, they simply burn it, and by burning that, they convert to electricity, which feed the sugar industry for their self-sufficient of electricity. But from my point of view as a chemist, what I feel that this is absolutely a waste of nature's precious material. Just we don't have the process, we don't have the technology to isolate these compounds in a commercial and physical way. Therefore, they just burn it out. So, and if you see, there is a TPC Tanzania plantation company in Canesco. It is one of the electricity producers in Tanzania. They have their report, and this is only 11% energy conversion. That means the total water that we convert is only the 11%, and 89% we are just wasting it. Let me just a little bit say about this. What is the hemicellulose of this material? They are basically made up of three components. One is hemicellulose, one is lignin, and one is the cellulose. This lignin is the outer covering of any plant cell. You see a plant cell, this is the outer covering is the lignin. The main core is the cellulose. The green lines you can see they are cellulose, and then these thread-like structures they are basically hemicellulose. Now this is the challenge over here. The challenge is this lignin, because this lignin is not very degradable easily, not by biological processes, not by chemical processes. Maybe the nature might have prepared these compounds in order to protect the inner plants from the microorganisms or the physical stress. So then this becomes a challenge, because when you want to extract these hemicelluloses and celluloses out of it, first you have to remove this lignin, and this lignin becomes a challenge for the industry, and therefore they are avoiding these processes, because the catalyst, few of them they have tried the catalytic studies, biological studies, but what has been found that the catalyst has been used in a reagent quantity than in a catalytic quantity, and then enhances the cost of production of this hemicelluloses and cellulose. Therefore, the industry is not interested in this. But there are a lot of research now going on in Europe, especially in Europe, and what we call this one as the biorefinery process, where we are refining not from the crude oil, but we are refining from a biological source, waste source, and then we are converting to an industrial physical compounds. So then it becomes a challenge how to remove the lignin and then extract these hemicelluloses, and then celluloses we can finally convert into bioethanol, and these hemicellulases can be sold to other industries, especially the paper and pulp industries, the pharmaceutical industry, as well as the ice cream industry, then to the biomedical applications. Now they're making a lot of nanotubes and all from these hemicellulases. So then this is the challenge. This is what bagasse that we have got. This is a waste product from the sugar industry. We got it from Tanzania Plantation Company, which is one of the biggest company in Tanzania, situated in Moshi, in Arusha region. So we take it that one, then we grounded that one, and after grinding, we further undergo studies. This is what we did, where we removed all the extractives from the sugarcane bagasse, which is the non-structural component of the plant cell wall. So plant cell wall consists of structural and non-structural components. So we remove this one by substituting all the extractives and non-polar compounds have been removed first by the turning in the mixture. So then we started isolating our products. First we did it with steam pretreatment at 200 degrees centigrade. Although this water extraction has also been done, has been carried out by many research groups. We also carried out this research group. We had a lot of publications in good journals like Bioresource Technology and all. But what we find that at 200 degrees centigrade for 10 minutes, it gives the optimum removal of lignin. Although not all, but we can remove at least 67 to 70% of lignin from the nucleocellulose material as far as bagasse is concerned. So then we extracted, cooled, and then it was precipitated, hemicellulose was precipitated in ethanol, and then when we call this one and dry it and we get this hemicellulose, we call this one the HST, means that steam treated hemicellulose. Then the residue that is left over from this steam treatment bagasse, we then treated that one with alkaline peroxide, and then we used 3% alkaline peroxide, and this is then our innovation, this magnesium sulfate. The concentration of the magnesium sulfate, if you vary that one, the percentage of lignin that comes out is also different. So we carried out a lot of experiments with this varying concentration of magnesium sulfate. That is again our different part of research, but here we are concentrating on the characterization of hemicellulose. So then we adjusted the pH to 11.6 with an average of 40 hours at 40 degrees centigrade for 12 hours. The extract was similarly concentrated, neutralized with SCl because we used an average over here, and then concentrated and precipitated in the same way as we did for the steam treatment.